There's a number that nobody in Washington wants to say out loud right now, and it is not a military number. It is $100. That is the price of a single barrel of oil as of this week, the highest it has been since the spring. And it did not rise because of inflation or supply chains or some distant economic cycle. It rose because a ceasefire that was supposed to end a war has just been rejected, again, by the one country that was never supposed to be able to say no. Iran said no. And somewhere between the Pentagon briefing rooms and the Truth Social posts, a question is starting to surface that almost nobody is willing to ask directly. What happens when the most powerful man in the world makes an offer? And the answer comes back refused. Here's what actually happened, stripped of the noise. Iran rejected a U.S. ceasefire deal Thursday presented to Tehran by the Iraqi prime minister, according to a New York Times report citing unnamed Iranian and Iraqi officials. And the Iraqi prime minister had visited Tehran days after meeting with President Trump at the White House the week before. This was not a phone call. This was not a tweet. This was a physical envoy, a head of government flying into Tehran with an American brokered offer in hand. And Iran still said no. Think about what that means for a second. This wasn't Iran refusing to pick up the phone. This was Iran examining the offer, weighing it, and rejecting it on the merits. Iranian officials said Tehran was not interested in a temporary deal that left the question of control of the Strait of Hormuz unresolved. Not the fighting, not the casualties, not even the strikes on its own soil. The Strait. Read that again because this is the detail almost every headline buried. The Strait of Hormuz is not just a strip of water. It is the single busiest oil choke point on the planet, a channel barely 2 miles wide at its narrowest point, through which roughly a fifth of the entire world's oil supply has historically passed every single day. Whoever controls the flow through that strait does not just control a regional dispute. They control leverage over the global economy itself. And Iran was not willing to sign anything that left that leverage in question, even temporarily, even as a goodwill gesture. That is not the behavior of a country that is out of options. That is the behavior of a country that believes it still holds the better hand. But here's where it gets worse. This is not the first ceasefire to collapse in this war. It is not even the second. On 28 February 2026, Israel and the United States launched airstrikes against Iran, killing its supreme leader and many other officials, destroying a large number of military and government targets and killing civilians. Iran responded with missile and drone strikes against Israel, U.S. bases, and U.S. allied countries in the Middle East, and by closing the Strait of Hormuz, disrupting global trade. In the aftermath of that opening escalation, Trump did not sound like a man looking for compromise. He wrote on 6th of March 2026 that there would be no deal with Iran except unconditional surrender. Sit with that phrase for a moment because it tells you everything about where this conflict started and how far it has had to travel to arrive at where it is now. A president who once demanded total capitulation is now, according to multiple reports, the one whose ceasefire offer just got turned down. To understand why that phrase landed the way it did, you have to go back even further to the ceasefire that started this entire pattern in the first place, back in the summer of last year. Trump announced that Iran and Israel had agreed to a complete and total ceasefire to be implemented in stages with an official end to the 12-day war to be saluted by the world. It sounded, for a moment, like history, except Iran's foreign minister denied any agreement on a ceasefire, and Iran's Fars news agency reported the ceasefire plan was entirely false and intended to divert public attention. Within hours, the ceasefire unraveled as both sides launched fresh attacks and accused each other of violations, and Trump told Israel's prime minister to call back planes headed toward Iran. Iran's president at the time struck a more measured tone, saying his country would respect the truce for as long as Israel did the same. Even then, in the very first version of this ceasefire cycle, the pattern was already visible: an announcement, a denial, a collapse within. Hours, and a fragile de-escalation held together by nothing more than a phone call. Everything that has happened since the February strikes, the April pause, the June collapse, and now this week's rejection is simply that same pattern repeating itself on a larger and more dangerous scale each time. And this is where everything changes, because between that unconditional surrender post and this week's rejection, there has been an entire hidden cycle of negotiation, collapse, and renewed fighting that most casual observers never fully tracked. In April, Trump announced a conditional 2-week ceasefire tied to Iran reopening the Strait of Hormuz, calling it a big day for world peace, with a 10-point proposal from Iran forming the basis for a potential long-term agreement. For a moment, it looked like the war might actually be ending. It did not end. Ceasefires in this conflict have a pattern, and once you see the pattern, you cannot unsee it. They get announced, they get violated within hours or days, they get blamed on both sides, and then they collapse entirely, only to be replaced by a new round of fighting that is somehow worse than the round before it. By June, that spring ceasefire had unraveled, and by July, Trump told reporters he believed the ceasefire was over following an exchange of attacks between the US and Iran. What most people are not seeing is how fast the escalation accelerated after that declaration. Within days, the US resumed its blockade of Iran and conducted airstrikes in the country for 12 consecutive days, while Iranian attacks on commercial vessels in the Strait of Hormuz continued. And what followed was not a skirmish. Now here's the part that should genuinely unsettle you, because it is not abstract. It is measurable in dollars, and it is measurable in blood. Nearly 100 US service members have suffered some level of injury, mostly minor, since July 7th, the Pentagon said, though CNN reported the military has been slow to disclose injuries during the conflict. This is not a hypothetical war being fought somewhere over the horizon, with no cost to the people funding it. People who left behind almost no physical trace of their existence, and yet who still shape at this very moment the immune systems of millions of people alive today, including a gene variant tied to survival at extreme high altitude, quietly inherited by populations living on the Tibetan Plateau. This is the strange, humbling truth that ancient DNA keeps forcing us to confront. We are not the clean, singular species we imagined ourselves to be. We are a mosaic, a patchwork stitched together from multiple ancient human lineages, some of which we have fossils for, and some of which, like our newly discovered ghost population, we may never find a single bone belonging to. We may only ever know they existed because their code survived when their bodies did not. So what was the secret hidden in that decade-old cave discovery, the one that reignited this entire mystery? As researchers dug deeper into the genome, comparing it against thousands of other ancient and modern samples in global databases, they began to notice something remarkable clustered around specific regions of the DNA, the regions responsible for immune function. It appeared that this ghost lineage carried a distinctive set of immune-related gene variants, ones associated with resistance to a particular category of ancient pathogens, and that these variants had been passed down. In other words, the descendants of an encounter that has otherwise been erased from history may be walking around right now with a biological gift bequeathed to them by ancestors whose names, faces, language, and culture are gone forever. A silent inheritance from a people who left absolutely nothing behind except this, the ability of their great-great-grandchildren's immune cells to recognize an invader a little bit faster than they otherwise would have. It is worth pausing here because it is easy for that sentence to sound like folklore, like something too poetic to be real. It is important to be precise about where also well documented, most famously in the case of a Neanderthal-derived immune gene variant linked to differing outcomes in modern viral infections. What remains, for any single new discovery, an open and active area of research, is the precise story of exactly which population contributed, which variant, when, and under what circumstances. Science does not deal in certainty so much as in steadily narrowing possibility. And the honest version of this story is not that scientists have solved the mystery completely, but that they have, for the first time, been able to see the outline of a mystery that used to be invisible entirely. But why would evolution preserve something like this at all? Why would a gene variant from a vanished population survive, generation after generation, for 10,000 years, rather than being diluted into nothing? The answer lies in one of the most elegant and ruthless mechanisms in all of biology. Natural selection does not care about sentiment. It does not preserve genes because they are interesting or meaningful. It preserves genes because they work. If a particular immune variant meant the difference between a child surviving a devastating epidemic and a child dying before the age of 5, then over enough generations, that variant would spread through a population like wildfire. Not because anyone chose it, but because everyone who lacked it eventually stopped having descendants. It is one of the coldest and most awe-inspiring truths of biology. Much of what we carry inside us today exists because it once saved someone's life during a catastrophe we have no written record of. This is where ancient DNA transforms from an academic curiosity into something with staggering real-world stakes. Because the very same technology that let scientists read a genetic diary written 10,000 years ago is now being used to read the diary each of us is writing right now, in real time, in hospitals and research labs around the world. The tool that made all of this possible is DNA sequencing, the process of determining the exact order of those 4 genetic letters across an entire genome. When the Human Genome Project completed its work in 2003, after 13 years and roughly $3 billion, it produced the first nearly complete map of human DNA. At the time, sequencing a single human genome took over a decade and cost a fortune. Today, that same task can be completed in a single day for a few $100s, a collapse in cost and time so dramatic that it has no real parallel in the history of technology, faster even than the improvements we saw in computing power during the rise of the microchip. That collapse in cost is what allows scientists to sequence not just one ancient skeleton, but thousands of them, comparing genomes across continents and millennia. Watching genetic makeup rather than treating every patient with the same generic drug at the same generic dose is no longer science fiction. In oncology, doctors already use genetic sequencing of tumors to identify which specific mutations are driving a cancer's growth, allowing them to select targeted therapies rather than blunt, one-size-fits-all chemotherapy. In rare genetic disease clinics, children who once went undiagnosed for years, sometimes their entire lives, can now have their exact genetic condition identified within days. And this brings us to a tool that did not exist when the Human Genome Project began, one that has changed genetics more radically than almost anything since the discovery of the double helix itself. CRISPR, short for clustered regularly interspaced short palindromic repeats, a name so clinical it almost disguises the sheer audacity of what the technology allows scientists to do. CRISPR was not invented in a lab from scratch. It was discovered, borrowed from bacteria, which had been using it for billions of years as a primitive immune system, a way of cutting up and remembering the DNA and then allowing the cell's own repair machinery to either disable a gene or insert a new one entirely. Their work earned them the Nobel Prize in Chemistry in 2020, one of the fastest transitions from discovery to Nobel recognition in the prize's history, a reflection of just how seismic this breakthrough truly was. The implications of that ability are almost dizzying to sit with. For the first time in the history of life on Earth, one species has developed the tools to directly and deliberately rewrite its own genetic code, and the code of nearly every other living organism on the planet. Gene therapy, once a distant dream, has already produced real treatments for previously incurable genetic diseases. Patients with certain inherited forms of blindness have had their vision partially restored. Patients with sickle cell disease, producing the defective hemoglobin responsible for their pain and organ damage, treatments that have received regulatory approval in multiple countries. A single-letter typo hidden in the vast library of the human genome, causing a lifetime of suffering. And modern science, for the first time, has a way to go in and correct that typo directly. But every door this technology opens also opens a difficult ethical question, and any honest telling of the story has to sit with that discomfort rather than rushing past it. In 2018, a scientist in China announced he had used CRISPR to edit the genomes of human embryos before birth, creating what were reported as the first genetically edited babies, altering a gene associated with resistance to a certain virus. The scientific community's reaction was swift and overwhelmingly critical, not necessarily because the underlying technology was impossible, but because editing the genome of an embryo changes not just that individual, but potentially every descendant they will ever have forever. And it was done without the kind of rigorous safety review, transparency, and international ethical consensus that such an irreversible act would demand. That scientist was later sentenced to prison in China for illegal medical practice. The episode became a defining case study in bioethics classrooms worldwide. A stark reminder that the line between healing a disease and permanently redesigning a human being is thinner and more consequential than most of us are comfortable admitting. This tension between astonishing capability and profound responsibility runs through nearly every corner of modern genetics, and it runs directly back to those ancient bones in the cave. Because the same sequencing technology that revealed our hidden ghost ancestors is also being used in an entirely different, almost mythic pursuit, the attempt to bring extinct species back from oblivion. Scientists have already sequenced surprisingly complete genomes from woolly mammoths preserved in Siberian permafrost, so well preserved in some cases that researchers have recovered nearly intact strands of ancient DNA, not just fragments. Using CRISPR, some research teams have begun inserting mammoth gene variants, the ones responsible for shaggy, insulating fur, thick subcutaneous fat, and cold-adapted blood, into the genome of Asian elephants, the mammoth's closest living relative, with the stated long-term goal of eventually creating a cold-tolerant elephant hybrid that could survive in Arctic tundra. This is not full de-extinction in the way popular imagination often pictures it, a mammoth walking out of a laboratory exactly as it once walked the ice. Age step. It is closer to genetic engineering inspired by an extinct species, a distinction serious scientists are careful to draw, even as headlines often blur it. Whether or not this work ever produces a living animal, and there is genuine scientific debate about whether it ever will. The ambition alone tells you how far this field has traveled from where it began. 100 years ago, paleontologists dreamed of finding bones. Now, some dream of finding a way to make those bones breathe again. And all of this, the ghost populations, the Neanderthal echoes in our immune systems, the gene-editing scissors borrowed from bacteria, the dream of resurrecting the mammoth, all of it eventually loops back to a much stranger and more intimate question. If DNA is simply a fixed code, an instruction manual written once and read forever unchanged, Then why do identical twins, who share exactly the same genetic sequence at birth, sometimes grow into adults with different disease risks, different mental health outcomes, different responses to stress, even different physical appearances as they age? The answer lies in a field that sounds almost paradoxical: epigenetics, the study of how genes are switched on or off without any change to the underlying DNA sequence at all. Think of the genome not as a script that is read the same way every single time, but as a script with certain pages taped shut, other pages highlighted in bright ink, some passages that a director chooses to perform loudly, and others that are permanently muted, all without ever rewriting a single word of the original text. Chemical tags, primarily a process called methylation, attach themselves to DNA over the course of a person's life, in response to nutrition, stress, trauma, environment, even the emotional experiences of a parent or grandparent, and these tags can influence which genes are actively expressed. Some of the most startling epigenetic research has examined descendants of populations who lived through severe famine or extreme trauma, finding altered stress hormone regulation and metabolic patterns in children and even grandchildren who never personally experienced that famine at all. The scientific community continues to debate exactly how much of this effect is passed directly through biological inheritance versus behavior, culture, and environment passed down through families. And this remains one of the most actively contested frontiers in the field, but the early evidence has already forced scientists to reconsider something that once seemed unthinkable: that the experiences of our ancestors, not just their genetic code, but the very traumas and triumphs they lived through, might leave a chemical echo capable of touching bodies not yet born. Suddenly, the story of that ghost population in the ancient cave takes on a different weight. It is not only their genetic letters that may have survived inside us. It may be, in ways scientists are only beginning to carefully investigate, something closer to an inherited chemical memory of whatever crisis they endured. A memory none of us can consciously access, but one that may still, in some small way, be shaping how our bodies respond to the world today. There's a reason this field keeps producing headlines that sound almost too dramatic to be real. And it is not because scientists are exaggerating. It is because the raw material they are working with, the molecule itself, is genuinely one of the strangest and most powerful things ever discovered by human beings. A single human cell contains roughly 6 feet of DNA, coiled and folded with such extraordinary efficiency that if you unwound the DNA inside every cell in your body and laid it end to end, it would stretch far enough to reach the sun and back, multiple times over. And within that 6 feet of molecular thread, in every single cell, is a complete copy of the entire instruction manual for building and running a human being, a manual written using an alphabet that has existed, largely unchanged in its basic chemistry, for roughly 4 billion years, since the very first flicker of life on this planet. Every living thing you have ever seen, every animal, every plant, every bacterium on a doorknob, is running some version of the same 4-letter code, endlessly copied, endlessly mutated, endlessly tested against the unforgiving arithmetic of survival. Forensic geneticists have learned to read this code in the smallest imaginable traces: a single hair follicle, a flake of skin left on a doorframe, a fleck of saliva on the rim of a glass, and use it to identify individuals with a precision that would have seemed like magic only a few decades ago, closing cold cases that had gone unsolved for 30 or 40 years by matching decades-old crime scene DNA against genetic genealogy databases built from ordinary people, voluntarily testing their own ancestry. The same fundamental technique, the same molecular alphabet, used to catch a killer, to diagnose a disease, to identify a ghost population from 10 millennia ago, to attempt the resurrection of an extinct giant, and to potentially rewrite the future of our own species, one edited gene at a time. Which brings us back, finally, to that cave, and to the people who lived and died there long before anyone thought to write anything down. We will likely never know their names. We will never hear the sound of their voices, or understand the words they used to comfort a frightened child or mourn someone they lost. Almost everything about who they were as individuals has been erased by 10,000 years of silence. But they are not entirely gone. A part of them, quiet and invisible, rides along inside the cells of people alive on this planet right now, walking down streets and cities those ancient people could never have imagined, encountering diseases those ancient people could never have named, protected in some small fraction of a percent, by an inheritance those ancient people never knew they were leaving behind. This is, in the end, the real story that ancient DNA keeps telling us over and over from every bone fragment and every frozen genome and every impossible ghost lineage we uncover. We are not separate from the deep past. We are not the end product of some clean, finished process. We are a living archive, walking around with fragments of extinct species and vanished peoples woven into the architecture of our own bodies, carrying forward decisions and survivals we will never be able to fully trace, written in a molecule too small to see and too powerful to fully comprehend. Every time a scientist cracks open an ancient genome, they are not just discovering the past, they are discovering us, a little more completely, one buried secret at a time. And somewhere out there, in soil we have not yet dug, in caves we have not yet entered, in ice that has not yet melted, there are more ghosts waiting, more secrets, more parts of the story of us still sealed away, still waiting to speak.